Okay, for all the many of you who are not here, welcome. To those sitting at home, I'm no doubt looking at this in week 13 or 14, but thank you for those that are here. Okay, let's start with Lepidoptera, and this is a pre-lab for tomorrow, so those of you here certainly will have an understanding of what we do in the lab a lot better. So it's going to be following a similar format which we use for all the major orders. So this is the first of the big polymetabolous orders that we're going to look at, and I'll introduce you. So first of all, Lepidoptera. Lepi is to do with scales, so it's called scaly wings, and you get them of anything from wingspans of sort of 3 millimetres up to 30 centimetres. So we've got quite a variation in size in Lepidoptera. Okay, Lepidoptera actually comprise two groups that you'll be fairly familiar with, moths and butterflies, but also another group known as skippers, and we'll elaborate what skippers are in a minute. Globally, we've got about 160,000 species described of Lepidoptera in 127 families. Again, like Hemiptera, we had about 100,000 described species in Hemiptera, so it's a bigger order, but we probably expect maybe not so many butterflies, because butterflies have been a subject of attention by amateur entomologists for many, many centuries, but certainly a lot of moths would be undescribed. So we might expect, again, double that might really exist out there in terms of Lepidoptera. In Australia, we've got 21,000 species and 82 families, and in Tasmania we have about 1,250 described species. That's nothing unusual about that, except in Tasmania most of our species are moths. We just have 39 species of butterflies and skippers, so we're looking more moth dominated down here, whereas maybe up north there's quite a lot more species of butterfly. Another point that comes from there is that I've said many species are known only from adults, so you might collect them at a light trap at night or flying around somewhere, but you have no idea what they are as a larvae or the food resources they feed on. So we might know 1,250 described species, but we've no idea what a lot of them actually feed on. It's quite a complex process to be able to rear things through and describe the caterpillars and the adults as well. So we're a fair way behind in understanding that. There are a number of different major groups. In Australia, moths are represented by 77 families, and we're dominated by one particular family, Oecophoridae. Mouthful, I know. 5,500 of our described 21,000 species are oecophorids. This is not typical of the rest of the world. Oecophorids are sort of leaf litter feeders, so they're found on woodland and forest floors, where they'll be feeding amongst the leaf litter in there. There are three other big families. Geometrids, about 2,300. Noctuids, about 2,000 species. We'll visit noctuids at different points in other lectures, but noctuids would typically be the dominant family around the world, and parrellids. Okay, so that's the moths. If we look at the butterflies, we have 4 families only of butterfly, and they include Papilionids, which are the classic butterfly, 18 species of those, and they would be, this is in Australia, like the Cairns birdwing, your classic picture of a butterfly. I'll show you that one in a moment. Pierrids, 32 species of them. They're the whites. Cabbage white butterfly is an example of that. Nymphalids, getting a few more nymphalids. They're the sort of oranges and browns. The wanderer or monarch butterfly is an example of a nymphalid that you may be familiar with. And lichenids, which we just saw in the video in between the two, the coppers and blues, and about 50% of those are associated with ants' nests in their immature stages. So we've got more species of lichenids than we have any other type of butterfly. Skippers are closely related to butterflies and the family Hesperids, and there are about 122 species of skippers. Okay, let's have a look at some of these in general appearance and take it from there. So here's a moth. We're familiar with moths. Moths are going to have quite a number of different variations on their antenna. We've already spoken about sexual dimorphism. They most frequently, with the exception of one group, and there are always exceptions to the generalisations, will be holding their wings back roof-like or A-frame-like, like a tent over the back of their abdomen, and they're generally going to have a fairly stout abdomen. Okay? We'll go through a few little pictures in a while on that differentiation. Butterfly, this is indeed the Cairns bird wing, so our butterflies are doing something different with their wings. They're holding their wings backwards over there, straight back over the back of themselves, and we can see they're quite different in that appearance. And then we have the skippers. Now this is the group that you may not have generally found in the common lexicon, but the skippers hold their wings at 45 degrees, so they're sort of halfway between what a moth and a butterfly is doing. And they also have an unusual looking antenna which looks like a little crochet hook on the end here of the antenna, which differentiates them from other groupings. So yes, a hooked like antenna. Okay, we want to now look at phylogeny and evolution. So where did Lepidoptera arise from, and when, and how long they've been about? Okay, so we've got our first fossils being later than the bugs. Remember, the bugs were about 240 million years ago. The first fossils we see are in the late Jurassic, about 160 million years ago. And some fantastic fossils in a paper that came out of China a couple of years back now. This is some of the illustrations of even the venation that you can see of the moths that came out of a particular site in China. So brilliant stuff. But near the end of the Cretaceous, 65 million years ago, they became a dominant group, linked to the evolution and diversification of the flowering plants. So the flowering plants, obviously, for flowers, they need pollination, so insects have exploited and vice versa for food resources and for pollination. So if we look at this chart that we see each time at the phylogeny and evolution, you can see here the Lepidoptera starting a little bit later, said about 160 million years ago, and then we can see this massive radiation once the angiosperms and the flowering plants, that is, started to really start to dominate in the landscape. So a little bit later, and not a significant rise like we saw with the Hemiptera, where they were fairly early on getting fairly large, capitalising on gymnosperms and pines and other things. When we look at it in terms of the phylogeny, again we revisit this slide each time, we can see here the Lepidoptera are most closely related to the Trichoptera or the caddisflies, okay, which is a group that some of you probably have collected, certainly at light traps and other things, which has an aquatic larval stage, and we'll visit them again in the course of the lecture. Okay, so now we're going to ask the question, well, what is a Lepidoptera, and how do we organise Lepidoptera into groupings? So, let's start with characteristics for recognising a Lepidoptera, and a few examples of people using Lepidoptera in various ways down below. So, the first one here is it's got broad scales on both surfaces of the wing. So they're broad, flat, hollow scales. It's an important thing that are on the surface of the wings. Now these scales actually enable a number of different things. They are helpful in thermoregulation. They're helpful in camouflage, if you need to camouflage. They have some help in enabling and assisting in flight, but they can also produce a lot of colours and variation in colours, which is quite dominant in butterflies in terms of communication between males and females with colours. So they're a nice palette or board for colours. So they're hollow, broad scales. Okay, they also have a coiled proboscis, which we're familiar with from looking at lecture 2, where we've got the maxilla rolled up. And they have something else here which is called an epiphysis, or a spur on the foretibia. So we'll get away from myself and Princess Eugenie and move on to some pictures of these two things. So here is your scales under scanning electron microscope, and more just under a normal microscope. Here you can see they're broad, they're flat, believe me, they're hollow, and if you angle those at certain positions, as we mentioned in the first lecture, you can get various diffraction and refraction to get metallic coloration and beautiful colours on wings. So those colours stay when they're pinned, which is why butterflies are quite keen things for collectors, because you can keep the colours quite well with most of the butterflies, because most of the coloration comes from orientation of scales. Now if we look at their close relative, the caddisfly, not terribly exciting looking order, but that's what a caddisfly looks like. It too has scales on its wings, but these scales are more hair-like, they're not broad and flat, and they're solid, okay? So that's the difference between the two, broad and solid. If we looked at the mouth parts of a caddisfly has chewing mouth parts, right? It doesn't have the coil proboscis. So if you're collecting and you're trying to tell me which order is which in your collection, have a good look at those things to make sure that you're not calling a caddisfly a moth, because that's a cardinal sin, getting your orders wrong in your insect collection. Here is that coiled proboscis or mouth part. Now we're going to see that quite commonly in a lot of insects, but Lepidoptera, but quite a few Lepidoptera have foregone any eating at all, so they don't have much mouth parts at all, and this is where they would have all the resources they require in their larval diet. So it's already there, they don't need to get any more food, so they're being able to feed, they don't need to feed in their adult life. But if they do have these mouth parts, they're going to look like that. The last one was this epiphysis, which you're always looking at me wondering what the hell's an epiphysis. I like to call it a comb, and it's on the foretibia. So if we look at this moth here, you can see a little epiphysis here. We look at it more clearly in magnification, we can see it here, here, and here. And what these do, we now know from the lecture we've just had that Lepidoptera are fairly keen on pheromones for communication, chemical communication. So one thing it does is it helps clean the antenna. So you've got, nothing stuck on your antenna to help with things. And the other thing it does is it helps clean that horrible long coiled mouth part. Okay, you've got to try and clean. Sometimes it's got stuck into something. So it's a cleaning comb, if you like, an epiphyseal spur. So we're looking for those three characters: the mouth part, which should typically be there, the epiphyseal spur, and the broad, flat, hollow scales. Okay, taxonomists are going to rearrange Lepidoptera into a number of different things, and I'm not so concerned about the names of these, but it's an interesting way of partitioning up some of the biology and looking at some of the key characters. So we have three suborders to deal with in Australia: Zykloptera, Aglossata, and Glossata. And as you can see there, Zykloptera has got 9 Australian species, Aglossata 1, and Glossata 21,000 plus. So nearly everything is in the suborder Glossata. But let's just have a look at these two groups, the Zykloptera and the Glossata. And a bit like we spoke about with Hemiptera, they're sort of like almost a Gondwana ancient group. That's sort of at the beginning of where possibly Lepidoptera began way back, 160, 120 million years ago. So we'll take a look at the Zykloptera first and see what they look like. So I'd love to see some specimens of these, but we rarely would. We have one species only in northwestern Tasmania, and they're all in this family, the 9 species called Micropteridae, and they're very, as I said, derivative early Lepidoptera. Done something totally different, so we're always going to get exceptions. They've got mandibulate mouth parts. They don't have a coiled mouth part, and they've got solid but flat wing scales. Okay, so something slightly different. What do they feed on? Well, in Australia, I don't think we know very much about them at all. But in New Zealand, they're found to be diurnal or day flying, and larvae have been found feeding on liverworts. So it's an interesting possibility that that's the sort of area that you would look for. Aglossata, we only had one species in Australia, and it's found in Queensland, so I don't expect you to find them, and even if you did see it, you'd probably think it was so boring you wouldn't want to pin it out, because it looks something like that. So it's got, again, mandibulate mouth parts rather than a coiled mouth part, and it's got solid wing scales, so just like the previous suborder, bit weird, bit old. Larvae feed within the seeds of Kauri pines, which is up in Queensland, and the adults are nocturnal, not diurnal like we thought with the other group. So very different, and Kauri pines are fairly high up in those Kauri trees, so larvae might be pretty hard to find. The adults look something like this. Okay, the glossata. The glossata is the big one, where everyone is actually there. We've got the coiled proboscis. Here we can see it on this hawk moth, very nice. And also here I'm drinking at the moment on that Pierid butterfly. They've got the hollow wing scales, and over 99% of all butterflies and moths are in the glossata. So they have the exactly what we're hoping for. For the mouth part is modified generally for sucking nectar or fluids. The hollow wing scales and everything else. There is a couple of other ways that taxonomists have tried to break up Lepidoptera. I've just mentioned those as curiosities rather than possibly important things. One of those is via the number of openings the females have. Either they can have one opening, which is for both laying eggs and mating, monotrizian, or they have two openings or a variation on that with separate apertures, which is for one for mating and one for copulation. Nearly most of them actually have two rather than one, so there is something interesting in that's going on, but we've got more than 94% of all glossata with two openings, one for mating, one for egg laying. The other way that they have tried to look at things, and it's hard to identify in some cases moths and butterflies when you're looking at venation because it's covered in scales. So we're not going to do adults so much just by sight in the lab, but we'll look at larvae because our larvae are the particularly important one for feeding and pest status. So, but we can have the venation either being similar, or homoneurous, between fore and hind wings, or different between fore and hind wings, and most of them are different. 98% of all Lepidoptera are different between fore and hind wings in venation. Okay, another term that you'll see around a fair bit in the common language, is probably not technically the correct terminology if you're an entomologist, but butterflies and moths are a common name. And these are a couple of things that help put you between butterflies and moths. The first one is the nature of the pupae. For moths it's sort of brown and smooth, for butterflies it's colourful and sculptured. But here we have a naked cocoon, whereas with moths it's either covered with some sort of silk, being either thin or thick silk. I'll come back to this slide in a moment, and I think it is on your handout maybe? No, it's not on your handout. So I'll come back to it, but I'll show you a picture there. So this is a butterfly pupae here, so you can see it's a naked chrysalis, it's just got one silk strand holding it on. This is a moth pupae, covered in silk, okay? Okay, we also have the time that they're active. Butterflies generally fly in the daytime, moths generally fly in the night time, but we do have day flying moths. We don't have night flying butterflies, but we do have day flying moths. The body is usually, the antenna of a butterfly is slender with a little knob on the end typically. Moths varies enormously with chemical communication. The body is heavy in a moth, light in a butterfly, bright coloured butterfly, dull coloured moth. And the wings we've already mentioned, either held like an A-frame over the back with a moth, or upright at rest with a butterfly. But I said there are always exceptions to these, so you put these together, you're probably going to get it right 99% or 95% of the time. But we do always find exceptions in these sort of generalisation rules that we have. Okay, immature stages. Let's have a look at immature stages. And immature stages are pretty important because if we think about it, one of the major things around Lepidoptera is damage to plants, to animal products, etc., food. So immature stages is often what you'll need to be able to identify, and even identifying, like we're trying to do in the lab tomorrow, whether it's a Lepidoptera, a Coleoptera, a Diptera or a Hymenoptera larvae is a beginning point. So how are we going to know that at immature stages is in fact the immature of a Lepidoptera? Okay, we've got a number of different things that we look at. So the larvae are generally called caterpillars and sometimes called grubs. It's not a term I like to use, we prefer to stick with caterpillars. But we have a number of different things here that we have, and I'll keep returning to this slide. The first one is prolegs. If you do have prolegs, not all have prolegs. If you have prolegs, they've got what's known as crotchets, which sounds very rude, but they're little hooks. So prolegs we mentioned in the first lecture on, no, the second lecture we had where we're talking about insect movement, or maybe that was the third lecture, insect movement, and we saw how they use prolegs for movement. So they're additional legs to the 3 pairs of thoracic legs. So if we look at this slide here, here's our caterpillar, and here's its 3 thoracic legs. Here are its prolegs. Remember, they're not jointed legs like the actual thoracic legs are. If we look at these prolegs, in this case it's got them on these segments, and we look at them under a microscope, we can see these little things called crotchets or hooks. So that's characteristic only of Lepidoptera if they have prolegs. Now prolegs are also present in some other orders, like say sawfly larvae may have prolegs too. So prolegs on their own aren't exclusively that of Lepidoptera. Okay, eye spots. We had a look at this in the lecture we've just done on visual communication in insects and communication in general. So they have 6 stomata. Remember, this is the visual organ of holometabolous insects. So when we look at that here, again, we have 6. So we're counting up eye spots. 1, 2, 3, 4, 5, 6. There's the 6th one there. 6 eye spots. Similar, if you could see there on this one here, number of eye spots. Not great for visual acuity, but good enough for something that's just going to sit and munch on leaves and other things, or be in burrows and tunnels. Okay, silk glands on the labium. So the labium is that bottom lip of the mouth parts. If you remember, we have the labrum as the roof, the labium as the bottom, and something known as adfrontal suture on the head capsule. So let's put those together in a slide and identify what they are and what you'll be looking for in tomorrow's lab when you're looking at recognising these things. So the first one is a silk gland. It's a little spinneret. Here we have the spinneret there. It's the silk, the external appearance of the silk gland. So there's a gland underneath, but the spinneret's just a tiny little thing. We probably wouldn't be looking for that, but the identification that there is silk doesn't just narrow it to Lepidoptera, but it excludes Coleoptera. Beetles don't produce silk, okay? Lepidoptera do, wasps can, there's a few things like that. Flies don't, okay? So we've got some narrowing if we're seeing silk of what it actually is, and there is a little spinneret there you can see. But the other thing is this adfrontal suture. So here we are looking at the head capsule head on, and there is a suture or fracture line we can see here. It's like a Y, an inverted Y, like a pair of jockey underpants or something, the other one upside down. So they actually fracture their head capsule on those suture lines. So when they molt, it'll fracture along that and that head capsule will peel off. So we've got this shape of a suture on the front of them. We won't see that on a beetle larvae, okay? Good way of getting them apart. Okay, let's take a look now at some of the aspects of the general biology of Lepidoptera. And what I want to pick on is just a few areas and say that we've already spoken about pheromones and communication and acoustics with Lepidoptera in the previous lecture, so I'm not going to talk about communication. And migration forms part of lecture 11, which, or 12, I think it's 11 or 12 on life history strategies, so week 6. We'll have a look at feeding. We'll have a look at predator defence. Remember when we're doing aphids where they were plumbed into something, or Hemiptera where they're plumbed in for a long time when they were in the Homoptera, then they needed ways of defending themselves. So we'll look how you defend yourself if you're sitting on a leaf feeding away, and look at some of these interesting relationships with other things like ants. Okay, so adults. So adults are not going to be, with 1 or 2 exceptions, the problem that you're going to face if you're getting damage. So they're really just interested in not nectar, or if you're some of those weird little suborders, the Zygloptera and the Glossata, Zygloptera and the other one, they're mandibulate species that can possibly eat pollen and spores. So nectar is what the majority are after, but we do get a couple of interesting things that happen in the butterflies, and there is a particularly interesting moth species that takes blood. So puddling does occur in butterflies. So they like to get some extras from their diet, so they want salts, minerals and amino acids. And they get this out of riverbed surfaces that might be slightly saline, decaying organic matter, etc., or drinking sweat from tears of insects. So I'll give you a nice picture of some of the puddling. and then go on to blood feeding with the Kelpie species. So here puddling, this is on a riverbed. You can see all these butterflies down there puddling away, picking up lots of salts and things that they need. Here is similar on a dead decaying skink, I think there, picking up there, and here they are in South Africa, puddling on dung of some large vertebrate. Vindula in Southeast Asia is a species that likes to drink sweat and tears. And there was a paper out only 6 months ago, identified a moth species as well, that in Madagascar. That here this bird's asleep at night and it's coming along and drinking from its eyes. Not taking blood, just drinking sweat and stuff around its eyes. So a little bit, but Vindula will do that more obviously in Southeast Asia. Now if you're a tapir in Borneo, then you might encounter this guy here, the vampire moth. Okay, so this is our blood feeder, and you can see the blood feeder has modified its maxillary pouch with nice little pointy barbs and things here to get in, and it drinks blood. So here it is on this person's hand trying to find its way in to drink blood from the finger. So you always get some interesting options, and that is blood feeding in vampire moths in Borneo. Okay, so let's get to where most of the action really is when we're talking about plants and protection and agriculture, which is larvae. So they feed externally, they feed internally, living or dead tissues of plant stems, leaves, trunks, flowers, fruit, root, bark, you name it. Any part of a plant, they'll be chewing on it. Okay? Rotting plants, leaf litter. I said the Oecophorids are the biggest family in Australia that are found in the leaf litter. We also have some that munch on fungi that might be growing in certain places, or on toadstools and mushrooms and things like that. And we have one family, the Tineids, the clothes moths that are amongst those, that feed on animal products like wool, hair, feathers, etc., which are the bane of people trying to store anything that might be made of wool, hair or feathers in their houses. So quite a lot of options they're feeding there, so we're going to get, you know, basically Lepidoptera as pests of a lot of those situations. I have a couple of weird ones that I just like to mention there. And there's two families there that are external parasites of plant hoppers and leaf hoppers and psyllids, so they sit on the outside of them, on the abdomen. And we've got another one here, another family, this family here, that actually is a parasite of leaf hoppers in its first instar, and then falls down and gets into ants' nests and acts as a predator in ants' nests. So some weird biology in there. We've got quite a few species, as you saw in that clip between lectures, that are ants, commensals in ant nests, and may even eat the larvae or pupae in those ant nests. The coppers and blues are those. We've got predation of eggs in 5 families here in Australia, and predation on scale insects in some as well, particularly those. There's a group in the Oecophorids that actually like taking scale insects that have fallen down on leaves in the forest underfloor and eat them rather than the leaves. So we've got a few little examples of parasitism and predation. And here is some of those here as examples to look at. So this is a geometrid that is a predator. So it's sitting there looking like a sit and wait predator, and the flies come along and it eats that. These are more exceptions than rules. And this is this one that in the Oecophorids that eats scale insects in the leaf litter. Okay, how are you going to not be eaten by something like a bird or parasitic wasp, taken by a parasitic wasp or another sort of predator? So defence. So there are two sorts of defence that you can have if you're like general in defence, but particularly if you're a caterpillar. One is what's known as a primary defence. So if it's a primary defence, what you're doing is you're avoiding being seen in the first place by a predator. Okay. A secondary defence is once you're seen by the predator, what the heck do you do? So crypsis is a primary defence, okay, with larvae and adults. And here you can see this one looks like a leaf, this one looks like the trunk of a tree, and this one looks like a twig, if you like. So they're all crypsis, so that A visually hunting bird, for instance, isn't going to necessarily identify that there is food there. If you're a moth in that family, Geometrid, one of the big families we have here, I'm sure you'll find Geometrids, which are the exception in that they have their wings out as moths, flat, not back over themselves like a roof. They often are fairly well camouflaged. So we have a Geometrid on the trunk here and a Geometrid on leaves. You can see quite a nice visual camouflage. Okay, if we're going to get the predator to actually have a go at us, it's seen us, what are some other options you can do for defence? One of those is you can apply toxins. And we have mentioned, I think, in the lecture on feeding digestion that plants have a number of toxins in them, and if you feed on a plant that's toxic, you can acquire those toxins as your own defence. Okay, so if you're toxic, you generally have a coloration that warns you. These are generally yellows, blacks, reds, whites. So the monarch butterfly is toxic. This is a classic experiment done early days on Apis imatisum and things where we've got the blue jay coming along and feeding, naive blue jay feeding on a monarch butterfly and throwing up afterwards. It's learnt, and then it's not probably going to take more monarch butterflies after that. If you're an entomologist, maybe there's a possibility of rabbit butterflies trying to stop you from getting them, but I don't think they've yet evolved. However, we do have mimics, and we've got a few mimics on display, possibly in this lab, certainly in our reference collection of different mimics. But in the Lepidoptera, we're looking at two types, Batesian and Mullerian mimicry. And Batesians, where a non-toxic animal mimics a toxic one. So here we have the monarch butterfly, that one that the blue jay tried and threw up on. And this is the viceroy butterfly, which is totally edible but mimics the monarch. So that's a Batesian mimicry. Well, Mullerian mimicry is where you might have lots of species that are all toxic, looking very similar, so they're enhancing that toxic effect. So that when a predator comes along, it's got a visual cue on that, thinks that I'm going to throw up if I eat that, so I'm not going to eat anything that looks like that, and they get a group effect by being all very similar. Helpful if you're not a very abundant species to do that, so that you will benefit from the abundant species. Okay, we could also suddenly try and startle anything like a butterfly, like a bird that's coming along to try and attack you. So we have quite a few moth species that have eye startles on them here, which they flash and it looks like eyes. And here is a caterpillar with the same effect, that it will throw up at you these eyes and suddenly think it's something bigger. This one hasn't yet evolved, but it would be an interesting option for butterflies. Something else you could possibly do is have hairs or of two types. One is you could just have really long hairs on you, a hairy caterpillar. You'll see quite a few of those. That makes it hard for, say, a wasp to stick its ovipositor in, or a bird gets a mouthful of hairs. Not so attractive, although cuckoos do apparently like hairy caterpillars, so everything has its little way around things. But cut moth caterpillars and quite a few caterpillars have what's known as urticating hairs. So they're almost like spines that have a little chemical on them on the end. So you go touch them and suddenly these things come out and they sting. So not just hairy, but stinging and hairy can be quite a good defence. We've already mentioned this one. Arctiids, the tiger moths, have that warning sound to bats that they're distasteful. But if you're getting attacked by a natural enemy, you can also silk, which is bungee jumping, say off the leaf. So you can see here the caterpillars jumped off that leaf, but then will be able to find its way up, and that wasp may therefore not attack it, but some wasps will actually rappel down the silk and attack it down there. So it's not a perfect strategy. Useful. The other one, of course, is you can get ants to attend yourself. We've mentioned that they're growing in nests, but you can also get they're protecting you while you're feeding. So over half of all blue butterflies, lichenids, are associated with ants, some living actually in the nest. So here's a beautiful tray of lichenids and butterflies, coppers and blues. This is the one that I want to mention to you, which is the Bathurst copper. which was an endangered species found in only certain regions which were being developed at the time. And they didn't know much about its larvae, but they've turned out to find out that the Bathurst copper actually is antitended and has a nice little gland that the ants like to take for sugary carbohydrates. And it only feeds at night. So what happens at night is that the ants shepherd it up at night and protect it, and then come and shepherd it back down in the daytime and goes back into the nest of the ants. So it gets up and feeds, gets the food that the ants want, and comes back. So interesting. Okay, let's have a look at economic impact. Now I've mentioned that, pest species, so I really want to just here try and sell you some good things about butterflies as much as bad things to begin with, and then we'll go on to the nastier side of it. So, first one, weed biocontrol agents. Quite a few Lepidoptera have been very successfully used for weed biocontrol. The most obvious and classic example of weed biocontrol, spectacular, was the prickly pear, Cactoblastis, which you can see all through here. Now in Australia in the 1920s there were large swathes of particularly southern Queensland and New South Wales that were absolutely covered in Cactoblastis, or prickly pear I should say, and they brought in a moth to control it, Cactoblastis moth, and you can see that this moth is too aposematic, so it's probably toxic. This is what the moth looks like. It's not very exciting, but within 6 months to a year it completely decimated all the prickly pear. So in the 1930s suddenly all this pasture and grazing land became available again. So impressed were they in those days with the scientific benefit of the control. There's a couple of monuments up in southeast Queensland area up there to Cactoblastis. So this is the Dalby monument to Cactoblastis and the agricultural benefit that it had. And this one is a town hall in Bunaga, which is known as the Cactoblastis Town Hall, that was built to celebrate the success of Cactoblastis as a biocontrol agent. That story is interesting. It goes a little bit further. That's actually got into parts of America now, and of course there are native cactuses in America. So, put in one place, somehow it's got into another part of America, and interesting to follow what's going to happen there. We mentioned this in the first lecture, that eating insects isn't really all that weird at all. It's quite logical. And there's a great little panel in Cullen and Cranston about mopane worms in South Africa. And they form a large part of the protein diet at certain times of the year for the people in that region. So you can see them all here selling mopane worms. This is quite big things. And they're looking at, there's a World Health Organization project looking at trying to culture these and increase the time frame over which that protein, which is essential for all sorts of things for people, increase it over a longer period of time than just the short window that mopane worms are there as part of the diet. Silk, also mentioned in the first lecture, silk production. Yeah, silk can be also a very expensive material that was produced, and it was first domesticated about 2,600 years ago. Oh no, 2,600 BC, sorry, so quite a long time back, over 4,000 years ago. And there's a lot of it in China and Japan, but a cocoon now, this is spinning the cocoon out from boiling water, but you can do that unravelling by machine now. But as an example for a little cocktail dress, so that someone like Audrey Hepburn might have worn, that would take about 1,700 silk cocoons to produce one dress. So you can see why it's quite expensive as an option. Okay, butterflies are always the one that people like. I said in the first lecture we have a schizophrenic relationship with butterflies. We don't like the immatures, but we love their adult forms. So we have butterfly zoos. This is Butterfly Zoo in Melbourne with artificial feeding, so people are very happy to go in there, and it's a good money generator. We also have some places, this is Costa Rica, where they're actually having butterfly farms for conservation purposes. So there are some very rare species of butterfly that people are very interested in collecting. So they grow the plant and the butterfly, harvest that, and then sell the butterflies on. and then reinvest that into conservation for the butterfly in a wider sense. So it's a win-win, if you like. But we do get some interesting things here. So this is a news article to November 2017, where 3 people were caught smuggling, and they were mailing these in to China, 2,800 butterflies into China, and some of those butterflies were selling for about $1,900 each. They're very rare and endangered species that people want, and unfortunately that seems to be part of human nature. Okay, we did say that adults aren't doing much, but there is one adult species here, the fruit-piercing moth, that has managed to become a negative. And it's found in parts of Southeast Asia and the Western Pacific Islands and Northern Australia. And we're looking at biocontrol agents, but with its mouth parts it's actually piercing fruits and damaging the surface of fruits. So it's been an ongoing program that they've been trying to work at to try and control it. So that's probably the only example I can think of adults being a problem. Pestitis generally comes from larvae. And unlike when we spoke about Lepidoptera, where we're talking about viruses and diseases, we were talking about aphids and most aphids being introduced. When we're talking about larvae and pests in Australia, we're talking about the majority of pest species being endemic. So we've gone and grown all these fantastic crops for them, brought these crops in, and they go, oh thanks, that looks good, I'll come across and eat that. Okay, so a lot of our endemic species that come across. And the key ones that have really liked crops are noctuids and paralids, but the hepialids, which you'll see these families tomorrow in the lab, are really big on pastures, and the tortricids, which are the leaf rollers, are really big in orchard situations. So all of these 4 families are big pestitis families in the Lepidoptera. Okay, so my best approach now for the last 10 minutes then is to look at some pest case studies and then take it from there. So I'm going to look at 1, 2, 3, 4, 5 different pest species, covering some of those families, the hepialids, the tortricids, noctuids, 3 of those families that I mentioned before. So the first one that I want to touch on is the hepialid, the corby grub. And those of you that are in pasture and dairy may be familiar pretty much with corby grubs. We have 2 species of corby grub in Tasmania. One known straight as the corby, which is only found in Tasmania, and then we have the winter corby, which is found in Tasmania and also southeastern Australia. So they're pests of pastures and urban lawns in all pastoral areas, but the winter corby tends to be in high rainfall areas like the northwest of Tasmania and parts of the mainland. So slightly different in distribution. The larvae live in tunnels quite deep down, 30, 40 centimetres down, and emerge only at night to feed on the grasses. So they're very hard to access to try and get control with, and I'll show you a slide of them in the next slide or 2. But they're very hard to access to actually kill if you want to control them. What they do like when they come up, they love ryegrasses, they love clovers, they love everything that we want to grow, you know, they don't like weeds. So they're feeding on everything that you might want in your pastures. So organophosphate insecticides, which are much more residual and much more toxic, have been used, but we're running out of probably good chemical controls for some of these sort of corbies. And because the Tasmanian one isn't a wide problem, it's more our own problem to try and deal with it. There's not any chemical company that's interested in, you know, corby on itself. It's just not a big enough market. So here we can see what the corby looks like. Hepialids, an interesting looking family there. So that's the adult. The Tasmanian only one, the corby, lays eggs in late spring. It's adult in late spring and laying eggs in January, March, and the winter's slightly phase shifted from that. Egg laying in November, December, so a little bit earlier. This is what the eggs look like, and you'll see those laid in areas where there are slightly longer grasses, where they'll lay and then they'll hatch and drop into the pasture. This is where the damage starts to come in for the corby. It's a larvae from April to October, but really damaging in August to October for the winter. the damage period is more towards the winter period, hence the call of winter corby. They look like this, they're not very spectacular or colourful, but here's corby right down in the ground, a long way down. You can see that's 40 centimetres down, and it'll come up at night. And if we look at the pasture after corby damage, we can see almost nothing left here. We can see the holes where they come up and down, and then you'll get weeds taking it over. This is a 20 cent piece here. Okay, light brown apple moth, so we're going to orcharding situations now. This is our own little unique animal, light brown apple moth too. It's a leaf roller, it's native, loves pome fruit, stone fruit, grapevines, but has quite a wide and catholic diet. It does damage leaves, but the main problem is feeding on the fruit, and the general method of control is pheromones. And I'll talk about that with a couple of slides. So this is what light brown apple moth looks like. If you've got it here, it's a leaf roller, so it's hiding underneath that leaf. Here's the pupae having pupated underneath that leaf, but it's fed all on the surface of that peach there. Here's the eggs. Here, the larvae, which have unrolled that to see it underneath because it's a leaf roller. The pupae. The adults aren't very spectacular. In fact, they're fairly boring. You can tell the sexual dimorphism. The males look like that, and the females look like this. But they're a very nondescript, not very spectacular looking moth that you wouldn't really notice that much at all. Interesting, the light brown apple moth got into California. It's in New Zealand, and it got into California a few years ago. So California's desperately trying to work out ways of controlling light brown apple moth and looking for natural enemies of it. But here we usually monitor with pheromone traps during spring and summer, and then when we see them we count physiological time or day degrees to when newly emerged larvae are there, because the larvae are vulnerable to pesticides when they're able to move around to find, but once they start to curl a leaf they're no longer really accessible, so it's in those very early instars that you can actually use a chemical control against them. Diamondback moth is a basket case all around the world. It's the most serious pest of any brassica crop, and chemicals have been prodigiously used against it, and there's been resistance throughout the world for all sorts of chemicals for diamondback moth. With those, the larvae are sort of leaf miners in their first instars and then feed on the external surface. And we have a couple of interesting things that are unique when we're talking about it in Tasmania, in that we have migrations of diamondback moth across Bass Strait. So we might have resistant strains from Victoria coming across the wrong times into here, and we're trying desperately to try and move away to more softer insecticides and natural enemies to control it. So it's pictures of what diamondback looks like here. This is a diamondback moth, that's why I call it diamondback. Again, it's not a very spectacular looking moth, quite a small moth. The larvae you can see here and the pupae you can see here. This is the damage, it's sort of like windows you can see, they don't feed right through. And you can tell them from cabbage white butterfly larvae, when they're young, the size of the cabbage white gets much larger than a diamondback by the amount of writhing or wriggling that they do when you poke them with something. And this is one of the biocontrol agents that's being used to try and control it. I've got two more species I want to talk about. Currant borer and armyworms to finish up. Currant borer is an interesting one. It's a beautiful moth. It's a clear wing moth. You'll see those tomorrow. So the wings are actually clear. You see the venation right through there. So really nice looking moth, but it's introduced. And what it's doing, we're not talking about feeding on leaves, we're talking about feeding in stems. So it's feeding in a stem, weakens the stem and breaks the stem if we've got winds and other things. So the problem again with this one is similar to the problem with light brown apple moth. Once it gets inside the stem, it's impossible access for control. So again, the same thing we're using. Pheromones is a common message with Lepidoptera, is that we were using a pheromone to try and identify when they're flying and then to time the spray so that when you've got these young instars that just hatch, you can actually access them and kill them before they get into the current. And there are quite interesting differences in blends depending on regions between New Zealand and Australia, and within regions in Australia in what works for the pheromone. So there has been some evolutionary change in pheromone blends in different areas. The last one here is armyworms. particular problem with pests of pastures, they're a noctuid. So we have two. We've got the common armyworm, and that looks like this as an adult, and the southern armyworm. These look very similar. Striping patterns and things are slightly different if you looked at them, but the adults look different. You can see here lots of differences. Now on this slide here, we want a little bit more. Again, we've got overwintering in Tasmania, but a lot flying across Bass Strait too here. And we have massive outbreaks. When they do come, we get massive outbreaks. So we had one in 2003 and one in 2007. And they feed on grasses, including, I should say, grain. So I've put that. But this is a small video of an armyworm outbreak up in northwest Tasmania in late 2017, just to give you an idea of why it's called army. Hopefully that works. Maybe it doesn't. So, but you can see lots and lots of. No, it doesn't want to work on that one. If you could see that, I'll go to that. Somewhere on this, maybe. Where is it? Armyworms. I'll try it here. There. There you can see just oodles and oodles of armyworms moving around there. So pretty terrifying, I guess, to have them coming through at any particular time. Okay, and the last one I want to show you here is a question I want to ask you. This is a newly described moth species. Any idea what you might describe that moth species as, looking at it?
Donald Trump.
Yeah, it is. It's this one here. Neopalpa Donald Trump eye. So, yeah, there is some sense of humour amongst Lepidoptera. Okay, thank you. We're done. See you tomorrow.